Hello everyone, good evening. Welcome to Aakash, BYJU'S NEET channel. I'm your teacher Ankita and I welcome you all in today's class where we will be discussing about the eukaryotic cells inside a very important system, and that is endomembrane system. So everyone, please make sure you stay with us till the end because we will be discussing this really very important topic that is very crucial for your NEET examination. Please make sure you're sitting with your notebook, your pen, your book, right? So that you can note on all the important things that we will be needing, right? And I hope that I'm clearly audible and visible to each one of you. Good evening, everyone. Good evening, Shalini, Jabba, Divya, Afas, Milinder. Yes, Afar, Captain Lucky, Drishtiman. Yes, good evening everyone. Hi Kishan. Hello, art and crafts. Good evening everyone. How are you all? All good? Am I clearly audible and visible to each one of you? Good evening, good evening. Thank you so much everyone for letting me know that I'm clearly audible and visible to you. Okay. Hello the runner. So are you ready everyone? Awesome. So let's get started. Hi, hello Paki. Hello. Okay. So everyone, in the last class, we had a discussion about the cell wall and the plasma membrane. I hope that all of you have watched that video. If you haven't, please make sure you go and watch that video where we discussed about the cell membrane and the plasma membrane. Okay. So here we have the homework question that we had discussed the other day. So the question, we had three questions. Question number 1, question number 2 over here, and question number 3. So of course over here, the options were not there, but of course you have written down the answer. So there are 2 students who gave up the correct answer. We have Milinder, right? Congratulations and thank you so much for, writing the answer, taking all the effort, and Mayuk. So congratulations, like you gave the correct answer, so very good. So the question 1, right, the plasma membrane exhibit an external substance. We know that the answer is option number D, it's selectively permeability, right? It actually tells us about its nature is selectively permeable. Then we have second question, endocytosis includes what? So of course we have phagocytosis, pinocytosis, both or none. So of course we know that it's both. It includes both the phagocytosis, eating, and of course drinking of the cell, right? Drinking, which is any fluid which is going inside the cell, and of course any food which is going. Okay. Then over here option number C is the correct answer. And in the accession, the reason we had a statement, all of the organelles of the eukaryotic cells are surrounded by either single or the double membrane, and of course we have in the reason they are stating that in the eukaryotic cells none of the cell organelles can work and exist without a membrane. We know that both the statements are absolutely incorrect. So option number C is correct. So I'm just writing over here, both the statements are false. Okay, remember that everyone. So everyone, are we clear with this? Right, so this was the homework question and we have our homework rock stars. Claps for them, pat on our back. Okay, so now everyone, are you ready for today's class? Good. Okay, so let's start everyone. In today's class, we will be discussing about the endomembrane system. Now, what is the endomembrane system? Of course, as a word we have, right, the endomembrane, we can easily figure it out what we have. Yes? Yes. Okay, give just a second. Captain Lucky, so what happens after the class, right? You can write in the comment section. If you go in the previous video comment section, you will be able to see the answers. Okay? So you can do that. Okay, so let's get started. So we're talking about the endomembrane as a word. Of course, we have endomembrane. What is the meaning of endo? We have discussed about the endocytosis. We have used this endo word, right? Endo specifically in a different occasion, different words. So it means the inside, right? So the inside membrane system that we have inside the cells, we call it as the endomembrane system. So we'll be discussing about it, right? When we talk about the endomembrane system, we know that it includes the very important thing, everyone. It includes the single membrane bound organelle. So the organelle that are there in the endomembrane system, All of these organelles have a single membrane. They are not double membrane, but they just have a single membrane. And of course, they are really involved in the packaging and the transportation of the material. It is absolutely absent in the prokaryotic cells, and it is absent in the RBC of mammals. Now, I'll not ask this question because we all know that why RBCs lack various organelles, because it can have more space for the carrying of the oxygen. So, this is a very general question that we have over here, right? So, we have the organelles which have single membrane. They plays a very important role in packaging, transportation. It is absent in the RBC, right? This is really very important for all of us. Now, let's understand what are the components that we have for the endomembrane system. Very important. So, here we have ER, Golgi bodies, lysosomes, and the vacuole. All of these organelles are single membrane, right? They are single membrane bound structure. Very important. ER, Golgi Apparatus or the Golgi bodies, vacuole, and the lysosome. So, this is clear? Yes. Collectively, we call all of it as the endomembrane system, right? And of course, endo, inside, within, right? So, of course, that is the reason why we have this interesting and super important name over here. Yes. Everyone, are we clear? Hello, yes, we just started. We're talking about, in today's class, we'll be discussing about the endomembrane system. And in endomembrane system, we have just talked about that we have 4 important components or the important organelles that we'll be discussing. We have ER, Golgi bodies, lysosomes, and vacuole. All of these organelles are single membrane bound structure. They just have a single membrane. They help in packaging, transportation of the material, right? And of course, they are making that also. And apart from that, RBCs lack these endomembrane. Basically, all these organelles that we're talking about. Okay. Good evening, Ayushi. So let's move, everyone. We'll be going into very quickly into the details of ER. Now, what you know about ER? Please do write in the comment section and tell me. ER is something that we have been studying, right? I'll always go back to our previous classes, because from there only we'll be able to get all the information. Ideally if we speak, right, there's nothing much added in this class 11. There are few hits, few lits, small bits and pieces of the topics which have been added over here. So we know that ER is a endoplasmic, right, endoplasmic reticulum. We know that it is of 2 types. RER, which is rough endoplasmic reticulum. It has ribosomes on it, and ribosomes help in the protein synthesis. We know that we have SER, which is smooth endoplasmic reticulum, which actually help in the synthesis of lipids. Now, proteins and lipids come together and form the plasma membrane by the process of membrane biogenesis. This all we know, right? Yes. Now, we know that ribosomes are absent in the smooth endoplasmic reticulum, right? And SER, along with the production of the synthesis of lipids, plays a very important role in detoxification of the cells, right? So we have these basic information, but let's understand what we have more over here that we will be learning in class 11. So here we go, and for our NEET examination. This is not mentioned in our textbook, but just an extra information for all of us to have, right? What is ER? We all know it's a very important organelle. First observed by the Garnier, right? And of course, the name was proposed, the endoplasmic reticulum. Name was proposed by the Potter in the year 1953. So remember this, right? This was a name, and of course, the name which was given. When we talk about the ER, right, we know that it's a single membrane bound organelle. It has a network of the tubes, right? It is a network of the tiny tubular and flat sacs-like structure. Okay, so we can see over here, this is the nucleus, and very close to it we have the ER. Yes, they are different person, Yash. Yes. Yes, Siddhi, I will recap. Just give me a movement. I will recap. You haven't missed much. We're just starting with the ER organelle. Okay. So, we know that endoplasmic reticulum is a single membrane bound structure. Of course, it has very tiny tube-like structure and cisternae that we have. Right. So, here we have. Now, when we're talking about the structure, right, we have cisterns or cisternae. Right. They're the long flattened structure, and they're arranged into the stacks like this. And we have tubules also over here. Now, these are irregular branch, and of course, they could be free or associated. Now if we have to make a nucleus, right? So if I just drawing it, so the ER, right, will start like this only. Write these flattened structure sometime. And of course we have these tubules also. Okay. So of course these are the tubular structure, and of course this what we have is the cistern. All of you, are you clear? Right, so these are the flat stack like structure. These are kind of this structure. Right everyone? Yes, and of course the tubule structures will be like this. Okay? So, we have two types over here. We have cisterns, flat stack like structures, flat structure which are kind of stack onto each other over here. Right? And of course these are the tubule structure that we have. Now, if you talk more further about, let's talk about the cisterns, and the tubules are very hollow. Basically they are hollow, and what we call that, when we have the empty space, I'm sure you would have seen a cylinder structure, right? So of course from outside it has a proper surrounding, but inside what we have, inside is a hollow structure, and that particular thing, the empty space that we have, is called as lumen. All of you are clear with this, right? Yes, Milendra, yes, it's there also. Very good. Everyone, I hope that it's clear. Absolutely correct. Now, we call it as a lumen. And based upon this space that we have, right, we know that ER can be divided into two parts. Two parts, I would not say two parts, but the two divisions that we have are intracellular space. So the space that we have, right, inside the ER and outside the ER, we have two very separate names for it. First name is the luminal, that is present within the ER. So this space, can you see? Let me just change the color of the pen, and just over here, can you see this space, green color I'm making? The internal space that is there inside the ER is the luminal. What space we have? We call it as the luminal. Whereas this space, can you see the outside space, this space? Outside the structure of the ER, right? We call this as the extraluminal. Okay, outside the ER, that is there in the cytoplasm. Two important space, extraluminal and the luminal. All of you, please make sure you remember this, right? So based upon these, we can have the question. Okay? Yes. Okay. I see that's a very easy question, and I'm sure your friends will be able to answer over here, and I will answer for sure. Yes, taken by the place is called as so could it be said like the place taken by the ER is called as lumen? No. The space, that is the place, the space which is there inside the ER, right, we call it as a lumen. Of course, we have a different, lumen remains same for other things also, okay, the empty space. But over here we have given them the name. All of you, are you clear with it? Quick thumbs up everyone in the chat. Yes. All of you, quick thumbs up. Jaldi say, all of you, please make sure you hit the like button for the video. Right? Very good. And in between, whenever you have time, please do take care of the screenshot, right? Okay. Chaliye. Now that we are clear with this, let's quickly discuss more further about the types of ER. So we have two types. We know that we have smooth ER and we have rough ER. Let's talk about the rough endoplasmic reticulum, and then we'll talk about the smooth endoplasmic reticulum. Here we have the images, everyone, you can see. See, we have the ribosomes attached onto it. We can see the red colors dotted balls right over here. These are the ribosomes, and over here there is a flat tube-like structure. So let's quickly discuss in detail about the ER, rough ER. So the rough ER is nothing but of course it has the ribosomes onto it. Hence of course we have the name. It is continuous. It's usually, can you see over here, this is a nucleus. It's a continuous with the outer membrane to the nucleus, right? It's starting from here. And of course the ribosomes that are present on the ER are very much involved in the protein synthesis, and of course, proteins are transferred to the ER lumen, right? So these yellow dots that I'm making, they are making the proteins, and these will be sent into the empty spaces. Everyone, are we clear? Right? So कहाँ पे बन रहे हैं ribosomes? Ribosomes कहाँ पे हैं? They are on the ER. ठीक है? ये लोग क्या करेंगे? ये proteins बनाएंगे, और ये proteins बना के जो empty space है ना, luminal space, जो space है वहाँ पे छोड़ देंगे, तो protein वहाँ पे आ गए. Clear? Are we clear with this? That is the rough ER. Okay? Now, we know that, see over here, we can have a picture image also. We have the ribosomes, right? Over here we have the ER proteins, and this is there in the lumen. Easy peasy. Yes. Now, let's talk about the smooth endoplasmic reticulum. They are continuous from the rough endoplasmic reticulum, we can see over here, right? They are not associated with the ribosomes. That's why we call them the smooth endoplasmic reticulum. They help in the synthesis of lipids. Apart from that, we know that they plays a very important role in the detoxification of the toxics also, right? So based upon their function, the number of smooth endoplasmic reticulum or the rough endoplasmic reticulum varies. For example, if we have a cell like pancreas, right, other cells where we have to synthesize more of enzymes, we will have which ER? Jaldi se all of you, what do you think about it? For example, if a cell has to produce a good amount of the enzyme, which ER they will have more? Smooth endoplasmic reticulum or rough endoplasmic reticulum? Very good. Yes, it's the rough endoplasmic reticulum. Short video is absolutely correct. Yes, Melinda, they come together, right? They release their synthesis, and of course, they actually help in the making of the plasma membrane, the membrane biogenesis. Very good. Very good, everyone. So we know that all of this is super, super important, right? Yes. Okay. Now, so here, everyone, this is something really very easy. You don't have to take a screenshot, but just have an understanding what is the main difference. Composed of cisterns, right? Cisternae. And it has more of tubules, synthesis, protein synthesis, and of course we have the lipid synthesis. This is important. ये हमें पहले से ही पता है, ज़्यादा इसमें trickier कोई भी चीज़ नहीं है. Yes. Now let's talk about from the ER what happens. So let's quickly revise the ER first. Now everyone, are we clear with this? All of you, quick thumbs up. I know कि आज का टॉपिक बहुत easy है, तो जल्दी से खत्म कर सकते हैं. बस I need your motivation. And I need you to be there with me and reply in the comment section. What is ER? We are talking about the endomembrane system. What is endomembrane system? Within or inside the cytoplasm system, right? So the endomembrane system, the endo word means within or inside, right? Now, right? So now what we have? Now we know that in the endomembrane system we have four important components: ER, Golgi Apparatus, Vacuole, and the lysosomes. First, we are starting with the ER. ER is endoplasmic reticulum. It is of two types, smooth ER and rough ER. Smooth ER have ribosomes and rough ER have no ribosomes. So rough endoplasmic reticulum help with the synthesis of proteins, and they are made up of stack-like structure, flattened stack-like structures, right? And they are kind of stack onto each other. Sorry, flattened structure which are called as cisternae, and they are kind of stack onto each other. Yes? Everyone, are we clear? Then we have smooth endoplasmic reticulum. They plays a very important role in the lipid synthesis. They are basically the formation that they have, right? The structure that they have is mainly of the tubules. They help in the lipid synthesis. They are present more in places where there's a detoxification of the toxin that happens. And both the proteins and the lipids come together and help in the formation of plasma membrane. Are we clear with this, everyone? Yes, very good. This is very easy. I hope that all of you got this part. We will be reading the NCERT, don't worry about it. We will read the NCERT also. Yes. Okay, we'll ask the question. So, now what we know that after the ER, the job is not done, right? The synthesis, the molecules that are made, the proteins and the lipid has to travel to different parts, right? Because of course there's a utilization of these. So we know that from the ER it will be going to the Golgi bodies, and we will see that happening in the next topic. So we know that proteins from the smooth endoplasmic reticulum and lipids from the SER will be transferred to the Golgi bodies in a vesicles, right? In the vesicles for the packaging and for the modification. Clear, everyone? Yes, we'll do that. Okay. Yes. Yes, absolutely. Okay, everyone, are we clear? How do ER forms Golgi bodies? No, they don't form that. We'll discuss that. Okay, everyone, are we clear? Yes, Charlie, the next part we will discuss is the Golgi bodies. But before that, एक बार हम पढ़ लेते हैं. Right? So we have the electron microscopes studies बताया कि there's a network of the reticulum, right? Tiny tubule structures scattered in the cytoplasm. We call them as the ER. Hence, ER divides the intracellular space into the two distinct compartments. Inside the cell, okay? Inside the cell that we're talking about. We have the luminal and the extraluminal space. The ER often shows ribosomes attached to it, and they actually help in the synthesis of proteins, right? And sometimes, of course, we call them as the rough endoplasmic reticulum. If the ribosomes are absent, we call them as smooth endoplasmic reticulum, है ना? उन्हें हम क्या बोलते हैं? Smooth endoplasmic reticulum बोलते हैं, और वो help करते हैं in the synthesis of lipids. SER frequently observed in the cells which are actively, right? जो बहुत ज़्यादा actively involved है protein synthesis में and secretion में, and they have extensive and continuous outwork, right? They are very close to the nucleus, of course, which is very much attached to it. And the smooth endoplasmic reticulum is a major site for the synthesis of lipids. And animals' cells, like lipids, actually help, right? In animal cells, lipids, like we have steroid hormones such as are synthesized in the SER. So all of these are really very important. हाँ, Datta noted your point. जहाँ पे ज़रूरत पड़ेगा, I'll speak in English and in Hindi, but we'll be trying our best to speak in English because you all will be writing the examination in English. समझाने के लिए I will use Hindi also. Everyone, are we clear? जल्दी से, I hope that all of you are clear with this. Okay, now let's take a look at the question, everyone. Which among the following extend from the nuclear membrane to the plasma membrane? Easy peasy, right? It's a very easy question. कौन है जो actually nuclear membrane से निकल रहा है और जाता है plasma membrane तक? Easy answer है, हमने एक ही organelle के बारे में पढ़ा है. So the answer is the ER. Very good. The answer is the ER, endoplasmic reticulum. So with this, everyone, we are done with the very first topic, right? Without taking much break, because अभी तो शुरुआत करी है, we have the energy in us. We'll move to the Golgi Apparatus. तो जो ER से सामान आ रहा है, राइट, वो कहाँ पे जाएगा? Golgi Apparatus, Golgi bodies में, ठीक है? अब अभी हमने क्या देखा? हमने देखा ये tubular structure, right? We have seen this tubular structure, right? ऐसे ऐसे tubular structures हैं. Just making a very just for our brief understanding, everyone. This is not how the structure looks, okay? Okay. So ऐसा structure है. We know कि proteins बन रहे हैं और proteins कहाँ पे हैं? Lumen में हैं, right? और इसी तरीके से lipids भी होंगे और ये Golgi Apparatus के पास में जाना है. How they'll be going? They'll be moving into the vesicles. Small vesicles में they'll get packed, right? And they will move to the Golgi bodies for the packaging, for the modification, and for the transportation. So let's understand. Golgi Parators, right? Class 9th में हमने पढ़ लिया था who discovered it and who gave the name, right? So it was discovered by the Camillo Golgi in the year 1898. उसी से नाम आया है. That's why we call it as Golgi bodies or Golgi Apparatus or Golgi complex. Usually in the plants we call it as the dictosome, right? Important term. इसे हम याद रखेंगे. The diameter, right? We have 0.5 micrometer to 1.0 micrometer in diameter. They're very, very tiny, right? And they lie really very close to the ER. This is nothing of any big thing. We all know that ER and Golgi Paratus are very close to each other because they have to work in sync. Now, this is important, everyone. Right? They are found in all the eukaryotic cells except, of course, the RBCs. Very important. Now, let's talk about the Golgi Parators. They are made up of the flattened stack-like structure. We can see over here, right? Flat, stack-like structure again. Flat है, उनको stack कर दिया है एक दूसरे के ऊपर. Consist of a disc-like cisternes, vesicles and tubules. Of course, यहाँ पे 3 modification है structure में. All of you, please do pay attention to this part. So they have 3 different types of little bit small modification into the shape, right? या फिर हम बोलेंगे पहले cisterns हैं, cisternes हैं, फिर vesicles भी हैं, and of course we will see the some of the tubular structures also. Okay, and these cisternes are stacked onto each other, which is very similar to the ER. So ये stack भी हो रखे हैं. सबको समझ में आ रहा है? Golgi Parators और ER का structure बहुत कुछ same है. Cisternes है, right? वो stack हो रखे हैं. Clear? And उसमें हम बहुत सारे variation देखते हैं. We have cisternes, we have vesicles and we have tubules also. Clear? मनीष बच्ची इनका सबका जो structure है वो different है, अभी हम देखेंगे. Clear? Yes everyone, are we clear? Yes. Okay? And ये जो हम देख रहे हैं cisternes, right? ये number vary करेगा Golgi complex में. एक सेल से दूसरे सेल में ऑफ कोर्स इट विल बी डेफिनेटली बी वेरी. ऐसा नहीं कि अच्छा यहाँ पे 3 है तो हर जगह पे 3 ही होंगे. नो, इट्स नॉट लाइक दैट. ओके, सो हियर वी हैव. नाउ लेट्स टॉक अबाउट मोर फर्दर, राइट? सो वी कैन सी ओवर हियर वी हैव द सिस्टर्न्स. एवरीवन सी दीज आर द वेसिकल्स एंड ओवर हियर कैन यू सी दिस ट्यूब्स, ट्यूब्यूल्स. मनीष आप पूछ रहे थे कैसा दिखता है स्ट्रक्चर. सो बच्चे हियर वी हैव सिस्टर्न्स, राइट? वी हैव flattened structures. Then we have vesicles and then we have the tubules. This is how they look. Okay. More further, in the Golgi Apparatus, right? There are 2 important phases. So हम हमेशा याद रखेंगे everyone. ER इधर है, यहाँ पे हमारा Golgi body है और यहाँ पे है हमारी plasma membrane. तो Golgi bodies के 2 phases होते हैं जो हमें हमेशा याद रखने हैं. जो फेस ER की तरफ फेस करेगा उसे हम क्या बोलते हैं? सिस. SIS, सिस. और जो प्लाज्मा मेम्ब्रेन की तरफ फेस करेगा उसे हम बोलते हैं ट्रांस. क्लियर? ऑल ऑफ यू आर वी क्लियर? यस? क्लियर एवरीवन? ट्रांस से आप क्या याद रखेंगे? ट्रांसपोर्टेशन, राइट? सिस से याद क्या रखेंगे? सिस्टम. ओके, क्लियर एवरीवन? So basically जो ER है, जब when it is sending the vesicles, right? जो vesicles बन रहे हैं, जो आ रहे हैं, they're going to the Golgi bodies, right? So which will be receiving them? The cis end will receive them. This particular end is also the convex or concave. Very good. It's convex, and of course we call it as a receiving end. Whereas the trans, which is facing towards the plasma membrane, is also called as the maturing form, maturing side also, and of course it has the concave shape. Remember this. Chaliye, now let's see this. So it has 2 phases, cis and trans. Cis phase face towards the endoplasmic reticulum, convex in shape, convex, right? Remember it like this, convex in shape, and it's the forming phase or the receiving end, right? It receives the vesicles from the ER, right? And whatever has been made, proteins, lipids, kind of come from the ER to the Golgi bodies in these vesicles. Okay? Are we clear with this? Then of course, we have the, 1 minute. Yes, we have the trans phase. It faces towards the cytoplasm, right? Towards the cytoplasm. Has a concave, it's concave in shape, and it's a maturing phase. Modified materials are packed and released from the trans phase from this side. Okay? Clear? So we have glycoproteins, glycolipids, all of these that are released from this particular side, trans phase. Okay? Everyone, are we clear? Remember, you know what? The question that usually comes, the PYQs that we have, and of course the important concept that we have is from cis and trans phase. Maturing phase बोल सकते हैं, maturing side बोल सकते हैं in the question. अच्छा, ये कौन सा phase है जो कहाँ पे है? Clear? So do remember the maturing maturing side, concave, trans, same. Cis, convex or receiving, same. Everyone, are we clear? Thumbs up everyone, are we clear with it? Yes, how many of you are here? Yes, very good. Yes, Milendra, Shalini, awesome. So we are clear with this part, right? Now let's talk about that how the things are happening. See where we have over here the labelings, right? We have the trans phase, cis phase. They've been transported from the ER and going towards the plasma membrane. Very good. Now here we have, chaliye dekhte hain. The vesicles from the Golgi Apparatus, they fuses with the vesicles, right? They fuse with the other vesicles for the storage, secrete out of the cells, and of course forms the lysosomes also. So we already know that the Golgi Apparatus plays a very important role in the formation of lysosomes. अगर lysosomes को अगर कोई आपसे पूछे क्वेश्चन में कि lysosomes कौन बनाता है तो आंसर क्या होगा? Golgi Apparatus, right? Clear everyone? So अभी तक हमने क्या पढ़ा? हमने पढ़ा Golgi के बारे में, Golgi कॉम्प्लेक्स के बारे में. हमने ये पढ़ा कि वो बहुत ज़्यादा क्लोज होते हैं ER से. उनका स्ट्रक्चर भी बहुत कुछ सिमिलर है किसके? ER से. सिस्टर्न्स होते हैं, सिस्टर्नस हैं एंड वो स्टैक हो रखे हैं एक दूसरे के ऊपर. 3 अलग अलग तरीके के स्ट्रक्चर्स हमें देखने को मिल जाते हैं. वी हैव द वेसिकल्स, सिस्टर्नस एंड ऑफ कोर्स वी हैव द ट्यूब्यूल्स आल्सो. 2 फेस होते हैं, सिस फेस टुवर्ड्स फेसिंग टुवर्ड्स द ER एंड देन वी हैव द ट्रांस फेस फेसिंग टुवर्ड्स द साइटोप्लाज्म और टू द प्लाज्मा मेम्ब्रेन. सिस साइड से रिसीविंग साइड है, कॉन्वेक्स इन शेप होता है और वहां से चीजें आएंगी वेसिकल्स में गोइंग इनटू द ER, सॉरी Golgi Apparatus. यहाँ पे पैकेजिंग होगी, मॉडिफिकेशन होगी एंड फिर दोबारा वो वेसिकल्स में आके अपनी अपनी जगह पे डिस्ट्रीब्यूट हो जाएंगे. क्लियर? ट्रांस फेज जो है वो हम हमेशा याद रखेंगे ट्रांसपोर्ट वर्ड से. इट इज ऑल्सो कॉल्ड एज अ ट्रांस फेज, राइट? मैच्योरिंग एंड एंड इट हैज अ कॉन्केव इन शेप. क्लियर? यस यस, शॉर्ट वीडियोस वी नो दैट के ER से ही तो प्रोटीन्स बन के आ रहे हैं और ये जो एंजाइम्स है हमारे हाइड्रोलिक एंजाइम्स बनते हैं प्रोटीन से. सो वी हैव एन इनडायरेक्ट कनेक्शन विद इट. क्लियर? ऑल ऑफ यू, आर यू क्लियर विद दिस? राइट? सो हियर वी कैन सी हाउ द मॉडिफिकेशन इज हैपनिंग. इजी पीजी एवरीवन, इजी पीजी. थम्स अप. सबको समझ में आया? हिंदी में भी हो गया, इंग्लिश में भी हो गया. क्विक थम्स अप एवरीवन. चलिए, अब ER हो गया है, Golgi Apparatus भी हो गए हैं आपके पास में. आई होप सब अपनी NCERT के साथ में है क्लास में. इसको जल्दी से पढ़ते हैं. NCERT क्या बोल रही है? ये बोल रही है कि किसने डिस्कवर किया? Golgi ने इन द ईयर 1898. कैम्लियो Golgi ने किया 1898 में. फर्स्ट ऑब्जर्व डेंसली स्टेन्ड रेटिकुलर स्ट्रक्चर नियर द न्यूक्लियस. Later of course we call them as the Golgi bodies after him. They are flat disc shaped sacs or cisternae of the 0.5 micrometers to 1 micrometer in diameter. They are stacked parallelly to each other. Varied numbers of cisternae are present in the Golgi complex. Okay, it's not fixed. The Golgi cisternae are, they are kind of arranged very close to the nucleus with the distinct convex, right, or the cis phase, which forms cis phase or the concave trans phase or the maturing phase. Maturing phase. Okay, cis and trans phase of the organelles are entirely different but are interconnected. They're kind of interconnected with each other. Yes, I will upload the notes. Yes. Now the Golgi Parators principally perform the function of the packaging material to be delivered either on the intracellular targets or to secrete outside the cell. सेल के अंदर और बाहर जो जाने वाली इंफॉर्मेशन है कौन मॉडिफाई कर रहा है, कौन पैक कर रहा है? Golgi Apparatus. Materials to be packed in the form of vesicles from the ER fused with the cis phase of the Golgis. ER से बन के आ रहा है, right? ER से बन के आ रहे हैं in a form of vesicles, fuse होते हैं हमारे Golgi Apparatus के साथ में. Then of course move forward to the maturing phase. यहाँ से बनते हैं, Golgis के अंदर जाएंगे, मॉडिफाई होंगे, पैक होंगे और फिर दोबारा वेसिकल्स में ट्रांस फेज से निकलते हुए साइटोप्लाज्म में जाएंगे एंड दिल बी गोइंग टू देयर डिफरेंट प्लेसेस जो रिक्वायरमेंट के हिसाब से. This explain that why the Golgi apparatus remain in a close association with the ER. इसलिए हमें समझ में आता है कि ER and Golgi bodies क्यों बहुत ज़्यादा पास में, क्योंकि उन्हें साथ में काम करना है. क्लियर? The number of the proteins synthesized by the ribosomes on the ER are modified in the cisterns of the Golgi Apparatus. ये भी important है. जो number of जो proteins जो बन के आ रहे हैं ER से वो modify कहाँ पे हो रहे हैं? Golgi Apparatus में before they are released from the transphase, and Golgi Apparatus is very important site for the formation of glycoproteins and glycolipids. We all know that they plays a very important role in the synthesis of lysosomes also. So everyone, are we clear with this? Done hai ye? I hope that this part is clear to each one of you. Right? We're done with the Golgi Apparatus. Now there are only two small topics that are remaining. We have vacuoles and we have the lysosomes. Awesome. Now everyone, let's take a look at the question that we have. Identify the packaging organelle of the cell. It's very easy question. I'm sure you'll be able to all of you will be able to answer this question. Identify the packaging organelle of the cell. We have ribosomes, Golgi complex, ER and vacuole, and the answer will be, some of you are saying C. Packaging organelle is what? Golgi complex, that is option number C. Very good. Awesome. That's a correct answer. So now everyone, we are done with the two important organelles that are there in the endomembrane system. Now we are left with two, which we have the lysosomes and we have the vacuole. So let's get started with the easiest one that we have. We have the lysosomes, right? And we call lysosomes as the digestive bodies, right? Lyso means digestive and soma means body. So they are the digesting body or the digestive body, inside which we'll see the digestion happening, breakdown of various materials happening. They're also known as the schizoid bags of the cell, right? And they were discovered by the Christine D. Devou in the year 1955. Remember the year, everyone. Okay. Now we all know that lysosomes are formed from the Golgi apparatus or the Golgi bodies. Now let's discuss about the important functions of it. They are formed by the Golgi bodies and they are present in all the animal cell except the exception, the RBC. Here we have, see, lysosomes. Then we have different types of lysosomes. We also call it as a polymorphism, right? Sorry, not the polymorphic. So what we know that the lysosomes, right, are at a very different stage altogether, right? So we know that they are single membrane bound structure. They are round in shape, sometimes very irregular, and they are filled with the hydrolytic enzymes. These enzymes are really very strong enzymes, like we have carbohydranase, protease, lipase. All of these are what? They will be digesting the carbohydrate. Protease will be digesting the protein. Lipase will be helping the digestion of lipids, right? And of course, they work in the high acidic condition, right? The newly synthesized, the newly formed lysosome is called as a primary lysosomes, right? Clear, everyone? This is called as a primary lysosomes. And we see that lysosomes have various different stages. So we call it as the polymorphic, right? Because they exist at a different stage altogether. They have a very different stages altogether, okay? Now we'll see about the other state. See, we have the hydrolytic enzyme over here. And these are the enzymes. You can take a screenshot of this, everyone. Just for our memory, for all of us to have, right? Please do take a screenshot. Short videos, of course, they have just been added into the vesicles. ER बना रहा है और vesicles में जा रहे हैं, फिर जा रहे हैं into the Golgi bodies. Yes. Thank you so much. Okay, chaliye. Now everyone, let's talk about the functions altogether of the lysosome, right? So intracellular digestion, inside the cell digestion, right? So we know that if something, be it the bacteria or the viruses are entering into the cell, we will see that what will happen, there was a formation right over here. Can you see? Phagosome. Yes, and as of course we will see it is getting combined with the lysosome. All of you, can you see this? Yes, right? So what we have is in the phagosome, basically there's a vesicle. Inside that we have the bacteria or basically the virus. Then when it fuses with the lysosomes, what we call it as? Right, we call it as the heterophagosome. Clear? So of course what happens, जैसे ही सिग्नल मिला कि कुछ फौरन आया है, what happens, lysosomes आएंगे, इसके साथ में फ्यूज हो जाएंगे and they'll start the digestion process. Clear? Good? Easy है, तो बहुत easy है. We have been discussing about the phagocytosis, right? Endocytosis similarly. But over here we have the addition of the lysosomes that we have. They will just release their digestive enzymes and that's how we'll see the breakdown of the material. Moving, we have the intracellularization. Along with that, they will actually help in the digestion of the food also. See over here, right? Food vacuole. Formation of the food vacuole. As the food will come, we will see the formation of the food vacuole. Then of course again the lysosomes will come. They will release the hydrolytic enzymes. They'll start the digestion process. And of course once it's done, we know that we have completed a small process which is super important for the cell. Right? Okay. Everyone, are we clear with this? Then of course, very important function, two important functions that we have. First of course, if any of the cell organelle is not working functionally, मतलब अच्छे से काम नहीं कर रही, if it's not functioning properly, what we will see, the lysosomes, right, will get into it. And of course, it will release the enzymes and okay, tata, bye-bye, right? It will not be able to survive from the harmful acids that are released by the lysosomes. And that's how it will break down. We'll see the breakdown of the organelle. Yes? Everyone, are we clear? Yes, all of you, quick thumbs up. So if they are breaking down the worn out cell organelle, we call it as the autophagosome. Yes? Or the whole process is called as auto. Right, of course, when it takes the life of their own cell also, similarly. Yes. Very good. Inflammation you're asking? I'll let you know. I hope that you're asking about the inflammation. Okay, chaliye. Similarly, everyone, we also call the side bags because of course they can digest the whole cell altogether. Right? Now let's talk about the extracellular digestion. Now this is super important, everyone. Right? Extracellular digestion. Extra. We have the osteoclast cells, right? ये आ सकता है एग्जाम में. किस cells में है? So these cells, right, releases their lysosomic content of the surface, right, on the surface of the bones, right? And this brings the extracellular deposition of the bones. Sorry, extracellular digestion of the bones. ये important question है PYQ. Clear? ये तो आ सकता है. कौन से सेल राइट अपना लाइसोसोमल कंटेंट रिलीज कर रहे हैं ताकि जो एक्स्ट्रा सेलुलर डाइजेशन ऑफ बोन्स होती है. आपको बहुत सारे एग्जाम्पल दे देंगे बट आपको क्या याद रखना है, क्या याद रखना है, कौन सा सेल है हमारे पास में, ऑस्टियोक्लास्ट. याद रहेगा ये. यस, ये कहां पे अपना डाइजेस्टिव जूस रिलीज करेंगे ऑन द सर्फेस ऑफ द बोन, उससे क्या होगा, डाइजेशन ऑफ द बोन्स होगा. This process also helps in regulating the level of the calcium, blood calcium. Clear? Everyone clear? इससे क्या हो रहा है, जो बोन्स है हमारी खराब होंगी, बोन का एरोजन होगा. सॉइल एरोजन पढ़ा है ना हमने, उसी तरीके से बोन का एरोजन होगा. Clear everyone? Thank you Milendra for helping your friends there. ओके चलिए, दिस इज इंपॉर्टेंट. आई हैव रिटन द PYQ, आ सकता है एग्जामिनेशन में. फिर उसके बाद में हम बात करते हैं सेलुलर डाइजेशन. अब अपने ही सेल को खुद ही डाइजेस्ट कर लिया. व्हेन द सेल इज नॉट परफॉर्मिंग वेल, राइट, इफ देयर समथिंग रॉन्ग विद द सेल, व्हाट विल द लाइसोसोम्स विल डू, दे विल रिलीज देयर एंजाइम्स, all the enzymes, and the cell will not be able to survive, and that is the reason why we call them as a suicide bags of the cell. इसे हम क्या बोलते हैं, ऑटोलाइसिस भी बोलते हैं. Clear everyone? All of you, are you clear with this? इसे हम ऑटोलाइसिस भी बोलते हैं. डन डन डन. तो लाइसोसोम्स तो एक्चुअली बहुत इजी हैं, ज्यादा कुछ दिया भी नहीं है, और जो इंफॉर्मेशन है बहुत इंपॉर्टेंट है. एक PYQ बताया मैंने, वो बहुत इंपॉर्टेंट है, प्लीज डू रिमेंबर दैट. So they are the membrane bound vesicle structure. They are formed from the packaging by the Golgi bodies, right? The Isolated lysosomal vesicles are found very rich in the all type of hydrolytic enzymes. We have the names. And of course, they work actively at the acidic pH. These enzymes are capable of digesting carbohydrates, protein, lipids, and nucleic acid. Yes, sometime, Elendra. Yes. Okay. Umen honey. Okay. Are we clear, everyone, with this? Chaliye. Here's a question for all of us. Which of the following is the function of the lysosomes? Digest food, cellular digestion, digest worn out cell organelle, or all of the above. ये तो बहुत easy है, right? This is super easy. Jaldi se all of you, please make sure you answer in the chat. Very good, very good. D, very good. The correct answer is all of the above. Easy peasy. Chaliye. We're done with this, everyone, and the last topic is here. I hope that all of you are excited, right? Everyone, I want to see a quick smiley in the chat. Yes, I hope कि आपको समझ में आ रहा है. I hope कि अब तक जो भी हमने पढ़ा है आपको clarity हो चुकी है. Hello, hello, hello Shanti. Yes. Okay, Captain Lucky Bharadwaj, I can see smileys. Yes, Shalini. Milrain, the vacuoles. Yes, absolutely correct. I think few of the students are here. Very good. A few have left. Okay, chaliye. Moving to the last topic, everyone, to the vacuole. Now, of course, vacuole, we know that occupies a central space in the plant cells. And of course, the number of vacuole in the animal cells are small, right? But they are more in number, but they're small in size. So, let's talk about the vacuole. They are very large and prominent in the plant cells, right? The membrane of the the membrane of the vacuoles is called as what? Tonoplast. Important word that you'll remember. And it occupies a 90% of the plant cell space, right? Very huge in the plant cells. They play a very important role and they occupy the central space. So here we have the vacuole. Now the other important point about this is that tonoplasts, of course, help the membrane that we have, right? The membrane of the vacuole actually help or facilitate the transport of the ions and the other materials against the concentration gradient. So this plays a very important role. Clear? This membrane. And it contains sap, water, and of course excretory products altogether. The concentration of the sap that we have, right? The concentration of the sap is higher than that of the cytoplasm. Let me write this over here, everyone. So, everyone note this point down. This is important. The concentration of the sap is higher than that of the cytoplasm. Hence, the concentration of ions that we have in minerals is significantly higher in the vacuoles as compared to the cytoplasm. Again, a very important point. Moving further, we know that, one minute, right? We have different types of vacuoles altogether, right? Alag-alag tarike ke vacuoles hai. One of the one that we're discussing now is the contractile vacuole. We have studied this in the amoeba, right? We have over here, yes, right, sorry, over here, the food vacuole. This is the nucleus, right? We have the cytoplasm, we have the pseudopodia, the false feet that actually help in capturing the food or engulfing the food, right? खाने को अंदर लाने में pseudopodia help करते हैं, जिन्हें हम false feet भी बोलते हैं, right? And we have the cell membrane. So food vacuole बनते हैं, yes, and it plays a very important role. ये contractile vacuole हो गए. Vacuole का दूसरा important function ये भी है कि वो help करते हैं किसमें? Osmoregulation में, और cells को बचाते हैं ताकि वो burst ना हो. Yes, here we have everyone. These are the food vacuole, and over here we have the contractile vacuole. Now they plays a very important role in the osmoregulation and in the excretion also. What is osmoregulation, everyone? Yes, what is osmoregulation? Water and salt ka balance, right? That we have in the body. So let's understand what is happening over here. So water will be entering due to the osmosis inside the cell. See over here, right? फिर क्या होगा? Excess water will enter into the vacuole, the contractile vacuole, right? That we have. उसके बाद में क्या होगा? It will swells. We can see there's a swelling of the vacuole, right? And of course, This will be moving to the edge and this will release the extra things, right? This will burst and will release whatever it's carrying. Expel the extra water. All of you clear? Yes, it plays a very important role in the osmoregulation. Yes, easy peasy. Then furthermore, right, and we know that the cycle will be repeated. We have the food vacuoles. In a case of amoeba, we have seen that, right? Here we have the food particle and here we have the food vacuole formation. We have the digestive juices, right, which are the lysosome, hydrolytic enzymes which are there, right, in the lysosomes. They will get attached to it. We have just seen, right? कोई food particle आया, lysosomes attach होगा, release करेगा digestive juices, break down होगा, and whatever waste will be released out from the cell. Okay? We have one more type, which is the gas vacuole, which are usually there, right, which are usually there in the prokaryotic cells that provides them the buoyancy, right? They can easily float around. मतलब float क्या? उसके अंदर वो वैसे से घूमते रहते हैं. Clear? Right? These vacuoles are floating. ऐसे. Easy peasy, everyone. All of you, are you clear with this? Okay, clear, crystal clear. So with this, everyone, we know that vacuoles are done. We cannot say that they're kind of absent, right? But we can say that they're present in a very, they are there, but they're very tiny in structure. Okay? Chaliye. That animal cells have small vacuoles because they don't have a need to store as much as water as the other organisms such as plants do. With this, everyone, we are kind of done, but I have few more questions for you, so stay with me. Some of the PYQs that we have. What is the name of the membrane covering the vacuole? What is the name of the membrane that is covering the vacuole? Yes, Gulzar. Milendra, okay. Tonoplast, very good. Option number C is the correct answer. Tonoplast is the name of the membrane that covers the vacuole. Here, everyone, we have the textbook screenshot, right? Vacuoles is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory products, and other material not useful for cell. The vacuoles are bound by the single membrane, which is called as tonoplast. In plant cells, vacuole occupy 90% of the volume of the cell. Easy. In plants, the tonoplast facilitate the transport of numbers of ions and material against the concentration gradient into the vacuole, and hence, their concentration is significantly higher in the vacuole than in the cytoplasm. Super important, everyone, this part. Then in amoeba, we have the contractile vacuole that plays a very important role in the maintenance of the osmoregulation and excretion, and in many cells like amoeba, we have food vacuole also, which actually help. Right? Food vacuoles are formed when there's engulfing of the food particle. Very good. Let's take a look at this question, everyone. Which of the following organelle is not a part of the endomembrane system? Now that we have discussed about the endomembrane system, it's very easy for us to find the answer for this question. Which of the following organelle is not a part of the endomembrane system? Mitochondria, ER, lysosomes and vacuole, and the correct answer is mitochondria. We know that mitochondria is not a part of the endomembrane system. So option number A is the correct answer. Here, everyone, few more questions for all of us. Jaldi se take a look over here. We have 3 more questions with us. Surface of Golgi. Let me just use a different pen. Surface of the Golgi, right, cisterns towards nucleus is called as what phase? And towards plasma membrane is called as what phase? Easy. What do you think, everyone? Which phase is towards the nucleus? We know that there's a phase towards the nucleus and there's a phase towards the plasma membrane. Cis, very good. Cis phase is towards the nucleus, right? And the maturing phase is there towards the plasma membrane. So option number D is the correct answer. Very good. See, this is how the questions can come. Just imagine, right? Looks very easy, right? Because now that we have studied it, we feel like, wow, it's a very easy question. So we have to focus on the important words which are given in the textbook. Next question, everyone. The Golgi Parators remain in a close association with the endoplasmic reticulum because material packed by Golgi Parators are transported to ER. Materials to be packed by Golgi apparatus body transported to the Golgi bodies from ER. Both Golgi Apparatus and ER have cisterns, and both Golgi Apparatus and ER possess ribosomes. Yes. Clear? Very good, very good. Yes. What is the correct answer, everyone? I can see a mix of B and D. Read the statement correct. Why the Golgi Paratus and the ER are there in a close association? Yes, option B is absolutely correct. Because the materials which are packed by Golgi bodies, right, transported to the Golgi bodies from ER. Golgi bodies कुछ काम कर पाएगा अगर ER उन्हें कुछ materials transport नहीं करेंगे तो? नहीं ना? That is really very important. Next question, everyone. ER divides the intracellular space into two distinct compartments. Luminal dash and extra luminal dash compartments. कहाँ पे होते हैं? Yes, Purvi. Everyone, yes, very good, very good. I know. जल्दी से everyone. So we know that we have the luminal कहाँ पे है? Inside the ER and outside the cytoplasm. So option number B is the correct answer. Very good, everyone. So with this, we are done, everyone. With today's class, here we have two questions for you. Right? We have question 1 and question 2. Please do write the answers in the comment section below. Right? These are the homework questions for all of us. I'll be waiting for your answers, everyone, in the comment section. Okay? With this, everyone, we are done with today's class. I hope that you have enjoyed today's class, right? Yes? Please make sure you hit the like button for the video. Do share it with your friends and subscribe to the channel if you haven't already, right? We will be picking up the pace, everyone, in the future. थोड़ा भी आराम से पढ़ रहे हैं ताकि थोड़ा सा अंडरस्टैंडिंग हो जाए किस तरीके से हम पढ़ाई करेंगे और क्या है important, right? So with this, everyone, I'll be leaving you. We'll be meeting really very soon. Do take care of yourself. Keep on reading the NCERT. क्लास कब हो गई है? NCERT पढ़ो. और जिन्होंने वो वीडियोस नहीं देखी हैं कि NCERT कैसे पढ़नी है, प्लीज मेक श्योर वो वीडियो जाके देखो. मैंने बहुत ही शॉर्ट तरीके में बताया कि हमें NCERT कैसे पढ़नी है. ठीक है, चलिए सभी बच्चों को गुड नाइट. लॉट्स ऑफ लव टू ईच वन ऑफ यू. अपना ध्यान रखें, हेल्दी रहें, राइट? अच्छा सोचें, मेहनत करते रहें और हमारे साथ पढ़ते रहें. बाय बाय एवरीवन. गुड नाइट.